Welcome back. This is another video in the sequence describing cell membranes. Today we're going to concentrate on the regions where membrane is made inside a cell, and then where membrane is altered in composition to match the destination or function of that membrane. Today we're going to concentrate on three different sets of enzymes that are present inside cells. These two work together, the flipases and the flopases, and at a different location we're going to discuss what happens with scramblbases. So I think it's best to talk about eukaryotic cells first, because it's easy to understand that location is very important as well as function of these enzymes. So let me draw a large cell, and we're going to use this large cell to understand geography as well as function. So let me label it as so we know we can compare the eukaryotic cell with a prokaryotic cell a little later on in the same video. So inside a eukaryotic cell we have many regions that are composed of membranes. So the nucleus, as we've discovered in the past, is made up of a double membrane. Then we have the endoplasmic reticulum, both smooth and rough, which itself is made up of a single layer of membrane. And then we have the Golgi apparatus, which is a stacked sequence of membrane structures that are represented in many books like a stack of bent pancakes. And we'll do the same thing in this video. So that's the Golgi. And then we have the destinations to which the membrane can be sent. For instance, you could send the membrane to peroxisomes, or which are vesicles, or to lysosomes, which are other types of vesicles. And there's many options that the cell has. So membrane can be sent to this location or that location, and indeed it can also be sent to the plasma membrane, as well as back to these other structures here. So the nucleus itself can be expanded and contracted in terms of its membrane by having new membrane targeted to that location. So let's go to the beginning. Where is new membrane inside a eukaryotic cell manufactured? And the answer is straightforward. It's always manufactured at the endoplasmic reticulum. The endoplasmic reticulum is lined with a single layer of a lipid bilayer membrane. So let's draw that in some detail. So what I'm going to do, I'm going to take this lower section of endoplasmic reticulum membrane and draw a larger cartoon here that will represent the two surfaces. So let's do that. So let's put a dotted line like this. So that represents a cross section of that. So we have phospholipid molecules such as this. Let's just draw 5 or 6. That will represent the inner surface. So the lumen of the ER is right here, and that faces these particular phospholipid molecules. And on the other side we have additional phospholipid molecules. Let's just balance them up. 6 on that side, let's put 6 on this side just for the sake of this exercise. And they would be facing this cytosol. So those are facing this region here, the cytosol, but not the lumen. The important thing for you to realize is that we're going to be talking about scramblbases first. So scramblbases are involved at the site of manufacture, which is here at the endoplasmic reticulum. The enzymes themselves are only present in the cytosol. So here's a scramblase. Here's another molecule of scramblase. There may be a third. These scramblbases are responsible for picking up lipid molecules, phospholipid molecules, and flipping them to the opposite side to balance the numbers. Why? Well, if we go if we go back step 1 to how lipid molecules are manufactured in the first place, food enters the cell. food molecules are then sent down biochemical pathways to make fatty acids and phosphates, and the composition of what the heads are. So the heads can be different. So some of these heads may be made of a green chemical. Let's put them as green like this. Some of the heads may be made of a substance that gives them a red property, like this. And some of them may be made of alternative types of a head molecule. Let's put them as orange. So it's important for students to realize that there could be up to 200 different types of phospholipid molecule present when lipid is made, depending on the needs of this particular cell. Okay. So these fatty acid molecules are combined together with the head groups by other enzymes that we're not talking about today. But those enzymes are also present in the cytosol. They're not present in the lumen of the endoplasmic reticulum. So let's put E for enzymes. There's many, many different types of enzymes, and their job is to manufacture new phospholipid molecules. So the only surface that's in contact with these enzymes and the newly made phospholipids is the outer surface. So in this diagram here, the enzymes will be present down here, these same enzymes that we spoke about here, and they would add phospholipids to this leaflet, the outer leaflet. So let's put that in here. So let's add a few more of these. There we go. There's one there. There's another one here. Another one here. These are newly made phospholipids which are added just to the lower leaflet, the cytoplasmic leaflet. Now you can see what's happening. The density of phospholipids in the bottom leaflet is increasing, and the density of the phospholipids in the lumen-facing leaflet stays the same. And what's going to become evident as we go forward is that the overpopulation of the bottom leaflet is going to cause the membrane to start bending. So the membrane will start bending like this because it has a far higher concentration of phospholipids there than here. And that tension is detected by the scramblelases. So the scramblelases, which are also down here, they detect that there's a distortion going on in the physical properties of this membrane, and then they come into action. And what they're going to do, they're going to non-selectively pick up some of these phospholipid molecules and flip them to the alternative side. So the word is non-selective. Non-selectively. That means they don't care which phospholipids they pick up. All they want to do is make sure that two sides are balanced in total number. So the scramblelases are located on the cytosolic surface of the endoplasmic reticulum, and their job is to balance the numbers of these phospholipids. So let's move some of these to the other side. So we'll take this one and we'll flip it to the other side. There you go. We'll take another one of these randomly because the scramblase doesn't care which one it picks up. It'll take this red one and it'll flip it to the other side, and we'll color that red and green accordingly. So that doesn't really matter. What eventually happens is that the two surfaces become balanced in absolute numbers once again, and the membrane loses its physical constraints, the tension that it built up earlier, and by moving stuff in that direction from the bottom leaflet to the top leaflet, i.e. from the cytosolic leaflet to the lumen leaflet, the two parts of the membrane, the newly made membrane, are balanced. And the identity of the phospholipids that make the top layer and the bottom layer, because it's a random process, will be the same. So they have a uniform composition. A new membrane has just been made by the endoplasmic reticulum by the action of a few select scramblelases. Once the membrane has been made, we now need to move our attention to another location within the cell, in this case the Golgi, because membrane is sent by being pinched off from the endoplasmic reticulum down the cytoskeleton to join the Golgi. So we're going to do that now. So let's get rid of some of the stuff that we don't need down here. And we're going to leave that diagram because we can use it again, because it's just moved from the endoplasmic reticulum to the Golgi. So let's get rid of all this other stuff. that we spoke about earlier. Let's get rid of these enzymes and the scramblase, because we now know where they function, only at the endoplasmic reticulum and only on one surface, the cytosolic surface. So let's turn our attention now to this membrane, the membrane belonging to the Golgi. So the Golgi does not manufacture a new membrane. That's left to the endoplasmic reticulum. What the Golgi does, it selectively conditions the membrane so that one leaflet is different to the other leaflet, so that one side can perform a different set of chemical reactions than the other side. And you may be saying, why? Well, as the membrane ends up in different locations, it needs to have a different composition of these phospholipids. So one thing to remember, A, is that different locations within the cell, different locations have different needs. Therefore, they have different chemistries. And the Golgi is responsible for manufacturing the right type of additional molecules to condition the phospholipids. So the Golgi knows which membrane to send where based on this. And the second thing the Golgi can do is it can put a type of asymmetry within the membrane itself, so that one surface is different to the other. So if you go here, the plasma membrane, the inner surface, the inner leaflet, may have different enzymes that are important in conducting biochemical reactions within the cytoplasm of the cell. The outer leaflet, facing the outside world, may have a different composition of phospholipids for that function. So it makes sense to have an asymmetry around the two leaflets. And I've said that enough, but the Golgi is responsible for doing that. So how does that happen? Well, unknown to us, but the Golgi knows which membrane is going to end up at which destination. So what it has are different enzymes just outside in the lumen around the Golgi. And those are the flipases and the flopases. So the flopases both start with an F, so that doesn't matter. So the F enzymes, here they are, they are very specific. So this flipase will be different to this flipase, which will be different to the others. This flipase will only pick up the red molecules and flip them to one side. So in the lumen of the Golgi, there are none of these enzymes. All the enzymes are out here in the cytosolic area. So this particular flipase, let's call it F1, the F1 flipase will pick up the red phospholipid molecules and move them to the other side. And it will do that as long as the enzyme is active. So it may pick up all the red ones or just a percentage of the red ones. It doesn't matter. So let's do that. So the red molecules are now flipped to this side. So let's assume that there are no red ones left on this leaflet. So that's what F1 does, and that's called a flipase. You may want to now move all the green ones to the other side. So there's another enzyme called F2. That may be a flopase. And what the flopase will do, it'll take all the green molecules and move them down to the bottom cytosolic surface. And it's very specific. It will not pick up the red ones because it can't, because of its shape and its active site. And it will move all these to this surface here. Okay, and these are the green ones. And there could be another flipase or flopase that moves the orange ones and balances their numbers out evenly between the two sides. So you can see that these different enzymes, they have particular roles in interacting with certain phospholipids. Eventually, you'll end up with phospholipids that are conditioned to be asymmetrically arranged between the two surfaces of the membrane. So the flipases and flopases, they work together to condition, like I said earlier, the asymmetry of this particular region of the lipid bilayer at the Golgi. Something different may be happening over here compared to over here, because you can change the composition and the presence of these enzymes by manufacturing and destroying them, as we'll learn in another lecture. So flipases 1, 2, and 3 may be active on this side, conditioning this membrane so it can join the peroxisomes. Another set of flipases may be over here. They may condition this so it can go and join the nuclear membrane. So this is how flipases and flopases function. They just alter the composition of the two leaflets of the membrane. While we're talking about enzymes, let's talk about the Golgi very quickly. Additional thing that the Golgi can do, it has enzymes on its interior surface, the lumen, and those enzymes are different to these enzymes. Those enzymes may add sugar groups to certain phospholipids, and because they're on the inner surface, they can only act on this leaflet. So they may add sugar groups like this to the heads of those phospholipids. And those sugar groups, if you follow another video, they have a particular orientation, and when they make their way to the surface, the sugar groups will only be found coating the outside surface of the plasma membrane, but those enzymes do not operate on this surface, and the inner surface of the membrane will not have those sugar groups. So the Golgi can modify the chemistry of phospholipids either directly or indirectly to condition it for the location at which it needs to do its job. So that's what happens in a eukaryotic cell. Now, how do we explain the existence of these same enzymes in the smaller prokaryotic cells? So let's go back, let's erase this, and let's go back and show you how that would work. So I need to get rid of most of this stuff, so bear with me please. So a great way to learn this is to compare and contrast what happens in prokaryotes and eukaryotes, and in many cases the biology is going to be similar but different, and understanding that difference in biology between prokaryotes and eukaryotes is a great way to learn cell biology. I need to get longer arms to do this, so I'm nearly there. I need to do some more exercise. All right, so I think we've got that bit. So we're just going to draw another cell, and this time it's going to be a prokaryotic cell, and then we're going to answer the same questions. Where is membrane made, and do these enzymes operate? So here we have a smaller cell. Pretend this is an E. coli cell. An E. coli cell only has one membrane. So it doesn't have an endoplasmic reticulum, it does not have a Golgi, and of course it doesn't have a nucleus. So the question then becomes, does it need to make more membrane? And the answer is yes, because E. coli cells do grow in length and width before they divide. So eventually this cell will divide in two and produce two daughter cells. That's another video. But for now, what we need to understand is, are these enzymes active? And if they are, where are they active? Well, as I just told you, the bacteria doesn't need to grow in membrane, so we would expect that scramblbases will be present, but where would they be present? Well, there's only one membrane, and the fatty acids and the heads of those fatty acids, just like in the previous example, are imported, and there's lots of biochemical pathways that lead to the production of those phospholipids, which look like jellyfish. Once those phospholipids are made, then they have to be inserted into the inner leaflet of the plasma membrane. So these are added to this side. And because they're added to the inner surface, the bacterial cell faces the same physical constraints that we had in the eukaryotic cell. The inner membrane becomes thicker at the expense of the outer membrane. In order to balance that, you need these scramblbases. So the scramblbases are located on the inner surface of the plasma membrane of bacteria. And what do? They non-discriminately flip some of these lipid molecules to the other side. Okay, like that. Therefore balancing the numbers, the absolute numbers between the two leaflets. Now in a bacteria, would you want some of the phospholipids on the inside appearing on the outside? And the answer may be no, it may be too dangerous. For instance, these phospholipids, maybe the green ones, only are present on the inner surface and never on the outer surface. So if you had a scramblase, the scramblase would be too general and flip over some of the green ones to the wrong side. And that may cause problems with the bacteria. So if you're following me, the logic suggests that you should not have any scramblelases, even though we said that they would exist if they were there. But once you go to analyze the question a bit deeper, they can't have any scramblelases, because the scramblelases will scramble the phospholipids to both sides, upsetting the asymmetrical nature of the membrane. So maybe bacteria don't have scramblelases as we know them. How about flopases and flipases? Get flip-flops? Yeah, the answer is yes, they do. So they have flipases and flopases, and what they do is they selectively move some of the molecules to one side or the other. Therefore, the inner surface of the membrane may have one type of chemistry, and the outer membrane will have an alternative type of chemistry. So please remember the location and function of these different enzymes in different types of cells. That's the end of this video, and the next video we'll look at membrane proteins. Thank you so much.